Today we are going to talk about positive inducible regulation of prokaryotic gene transcription. Prokaryotes regulate their genes through transcription. This transcription of genes occurs at a segment of the DNA known as the operon. The main structures of the operon that we'll be focusing on are the promoter region, the operator, and the structural genes. Structural genes code for proteins, which are then used for cellular processes such as metabolism. In order for these genes to be coded, RNA polymerase must bind to the promoter region and travel along the DNA strand past the operator and through the structural gene region. The operator, however, can block the RNA polymerase from traveling to the structural gene region depending on whether or not a regulatory protein is bound to it or not. In the case of positive inducible regulation, the regulatory protein that binds to the operator is known as an activator protein. Once the activator protein binds to the operator, transcription is stimulated. However, in order for the activator to bind to the operator, a small molecule must first bind to the activator protein. This is known as inducible control. Now that the activator protein is activated, it may bind to the operator and subsequently activate it. Once the operator is activated, RNA polymerase may travel past it and stimulate the structural genes to be transcribed for gene expression. Let's take a look at how this process occurs by using a simple analogy. In this positive inducible analogy, we will be using my stubborn hound, Henry. He is a very good boy, but does not listen to commands unless he is given a treat. Two things are required in order for Henry to come to me: a command and a treat. In this analogy, transcription is the action of Henry coming to me. The activator protein is the command come, and the small molecule or effector is a treat. In this first video, we will see what happens when I command Henry to come to me without a treat. Henry, come. Come. As we can see, in the absence of a treat, Henry will not come to me. In other words, in the absence of the effector molecule, the command or activator protein is not able to stimulate the transcription of the action of Henry coming to me. In the next video, we will see what happens when I command Henry to come to me with a treat. Henry, come. Good boy! Good boy! This time, the effector molecule is present and can therefore stimulate the activator protein, or the command, to stimulate the transcription of Henry coming to me.